Welcome back to the Deep Dive. Today we are getting into the absolute fundamentals of life. We're talking about macromolecules.
Right. Carbohydrates, proteins, and lipids.
And we're not just defining them, we're going to unpack the ingenious chemistry we use to even spot them in the first place. And then we'll use that knowledge to understand something incredible like spider silk.
Our sources today are a mix, really. Part practical lab manual, part advanced biochemistry text. So our mission, you could say, is to connect a simple color change you might see in a test tube to the stunningly complex 3D architecture of a protein.
Okay, let's unpack this then. We're focused on how a little splash of color can tell you what's there, and then how the structure of these things gives them these just astonishing functions.
It all starts with something called an indicator.
A kind of molecular spy.
I like that. Yeah, it's a chemical that changes color when a very specific reaction happens. But you can't just trust it blindly. Before any test, you need to run your controls.
And this is the non-negotiable part of any lab work,
Yeah.
right? The controls.
Absolutely. They're your insurance policy.
So you have two. First, the positive control.
Right. This is a sample where you know the molecule you're looking for is present. So if you're testing for sugar, you use a known sugar solution.
And why is that so critical?
Well, because if your positive control fails, if that known sugar solution doesn't give you the color change you expect, then something is wrong.
Your reagents might be bad, your heater's not working.
Exactly. And if that fails, all of your results for every other sample are completely invalid. You have to throw them all out.
And the flip side is the negative control, which is...
Usually just pure water. It has none of the molecule you're testing for. This shows you what a negative result is supposed to look like. If it changes color, you've got contamination somewhere.
So those controls really anchor the entire experiment.
They do. Okay, so let's apply this. First up, carbohydrates. We're talking molecules made of carbon, hydrogen, oxygen, usually in that 1 to 2 to 1 ratio we all learned.
Like glucose. And we have, the simple sugars, the monosaccharides.
Then disaccharides, which are 2 of them linked together, like table sugar, and polysaccharides, which are the long chains, like starch.
So how do we find just the simple ones, the monosaccharides?
For that, we use the Benedict's test. And the key here is understanding why it works. Monosaccharides are what we call reducing sugars.
Meaning they can donate electrons?
Precisely. So
Yeah.
you take your sample, you add the blue Benedict's reagent, which is full of copper ions,
Yeah.
and you heat it.
To about 90 degrees Celsius, right?
Yep. And at that temperature, the sugar donates electrons to those copper ions.
Yeah.
That chemical process, the reduction, changes the copper state, and it changes its color.
From blue, and then it goes through maybe green if there's just a little bit of sugar.
All the way to a thick orange or brick red color if there's a lot of sugar present.
And the really important part is that the more complex sugars, like starch, they don't react.
They generally don't, no. The reducing groups are tied up in the bonds holding them together. So this test is a fantastic shortcut for telling simple sugars apart from complex ones.
I love that. That structural difference is everything. Okay, on to proteins. They're polymers of amino acids, all joined by peptide bonds.
And to detect those, we use the peptide bond test, sometimes called the Biuret test.
This one uses 2 chemicals, potassium hydroxide, KOH, and copper sulfate.
Right. Now, we should be really precise about what the KOH does. It doesn't actually break the protein apart.
Oh, okay. So what is its role?
It creates a highly alkaline environment, a basic pH. And that specific condition is what allows the copper ions from the copper sulfate to form a special complex.
With the peptide bonds themselves?
Specifically with the nitrogen atoms in the backbone of those peptide bonds. When that complex forms, the solution turns this really vibrant purple.
So more protein means more peptide bonds, which means a darker purple.
A much darker purple. And because things like free amino acids or lipids don't have that specific bond structure, they won't react. It's very specific.
Okay, this brings us to a perfect real-world application. Let's talk about the saga of the soda dispenser.
Ah, yes, a classic problem.
The situation is this. A server messes up the lines. Regular cola, which is full of high fructose corn syrup, is now coming out of the diet tap. And diet cola is in the regular tap.
Which for a diabetic customer isn't just an inconvenience, it's a genuine health risk.
A huge risk. So how does this basic chemistry we've just talked about solve this high-stakes problem?
Well, fructose is a monosaccharide. It's a reducing sugar. The artificial sweeteners in diet soda are not.
So you grab 2 samples from the machine, run the Benedict's test.
Add that heat, and the regular soda, all the fructose, will immediately give you that bright orange positive result. The diet one will stay blue.
And just like that, you know which line is contaminated. You've prevented a potential medical emergency and saved the business from throwing out all its product.
It's amazing, isn't it? A multi-million-dollar industry safety net comes down to a few cents of copper sulfate.
It really is. Okay, let's quickly cover our third group, lipids.
Lipids. The defining feature here isn't a repeating subunit, but a property. They're not soluble in water. They're nonpolar.
We're talking about fats, oils, waxes, made of glycerol and fatty acids.
And because they don't dissolve in water, we can't really do a simple test tube reaction. We have to use a different approach. We use a stain called Sudan IV.
Right. And the lab manuals are very clear. Wear gloves. It's a possible carcinogen.
Definitely. Safety first. So the principle here is like dissolves like. Sudan V is a nonpolar stain. Lipids are nonpolar molecules.
So you'd spot, say, different milks or oils onto a piece of filter paper and let it dry.
Exactly. Then you soak the paper in the Sudan V solution, and then you rinse it with water. The water washes the stain off the plain paper, but
Wherever there was lipid, the nonpolar stain sticks to the nonpolar fat.
and you're left with these red spots. And you can see, just by looking, the difference. The spot from heavy cream will be much, much darker than the spot from skim milk. It's a simple but very effective visual test.
Okay, so we found them. Now we have to make a big shift in our thinking. Because with proteins, it's not just about whether they're present, it's about their shape, their architecture.
Yes. Proteins are the ultimate molecular tools, and their power comes from their incredibly complex three-dimensional structures,
It all starts with the primary structure.
which is just the sequence, the specific order of the amino acids linked by those peptide bonds.
That's the blueprint.
It dictates everything. From there, that chain starts to fold into local repeating shapes. That's the secondary structure.
Things like the alpha helix, which is like a coil, and the beta pleated sheet, which is more like a folded fan.
Right, and those are held together by hydrogen bonds along the protein's backbone.
And then the whole thing folds up into its final functional 3D shape, the tertiary structure. But what holds that shape together? It seems like it would be so fragile.
It is. But there are powerful forces at work. And the main one, the biggest driving force, is actually the hydrophobic interaction.
The water-fearing effect.
Exactly. The amino acids that are nonpolar, that hate water, they all rush to the inside of the protein. They try to bury themselves in the core, away from the watery environment of the cell.
Why does that stabilize the protein?
It's really about the water. By clumping together on the inside, the nonpolar groups allow the surrounding water molecules to be more disordered, which is a more favorable state. It's a huge driver of the folding process. We also get some stronger bonds, like disulfide bridges, which really lock the structure in place.
But it is fragile. If you disrupt those weaker bonds, the whole thing falls apart.
That's denaturation, right?
That's denaturation. And it's important to know you're not breaking the primary sequence. The peptide bonds are still intact. You're just unfolding it, disrupting the secondary and tertiary structure.
So heat, strong acids, even just whipping egg whites.
All of it. It's all about adding stress that those weak interactions can't handle.
The egg is the perfect example. You have this clear soluble protein, egg albumin. You heat it, and it becomes this opaque, insoluble solid.
And that's usually irreversible. You've unfolded it so much it can't find its way back to its original functional shape.
It just shows how precise this all has to be, which finally brings us to the most amazing example of protein structure. Spider silk, biosteel. It's just an incredible material. The sources are clear on this. Dragline silk is, by weight, about 5 times tougher than high-grade steel.
5 times. And twice as tough as Kevlar. I mean, that's just mind-boggling. How does it get that strong from just a chain of amino acids?
It's all in the folding. Spider silk protein is mostly made of 2 amino acids, alanine and glycine.
Okay, so what do?
The stretches that are rich in alanine fold up into those beta-pleated sheets we mentioned, and they stack together perfectly to form these hard microcrystalline blocks.
So that's where the strength comes from, the resistance to being pulled apart.
That's the tensile strength. But if that was all it would be strong but brittle. It would just snap.
But spider silk is incredibly elastic.
And that's where the glycine comes in. The glycine-rich parts don't form sheets. They form these amorphous random coils. They're like molecular springs connecting the hard crystalline blocks.
So it's a composite material. You have these hard, strong blocks for strength linked by these stretchy, elastic sections for flexibility.
It's a masterpiece of material science, all encoded in a single protein sequence.
But wait, if heat and stress denature proteins, how does a spider turn a liquid protein solution in its gland into this solid, super strong fiber without, boiling it.
That is the marvel of the spinning duct. Inside the spider, the silk protein is a highly concentrated liquid, mostly in that alpha helical state. As the spider pulls it through the duct gets narrower and narrower.
So it's applying physical force, sheer stress.
A lot of it. That forces the molecules to line up. But at the same time, the spider is actively changing the chemical environment inside the duct. It pumps out certain ions like sodium and pumps in others like hydrogen ions.
So it's making the solution more acidic.
Yes, and that combination, the physical stress and the drop in pH, is the trigger. It forces the protein to instantly flip from the soluble alpha helices into those insoluble hydrophobic beta-pleated sheets.
Which forces the water out and solidifies the fiber right as it exits the spider.
It's a biomechanical process we still can't perfectly replicate.
And that's the big challenge, isn't it? The applications for this stuff are endless. Surgical thread, lightweight armor, but we can't make it.
We can't farm spiders. They're cannibals. And even when we engineer bacteria or goats to produce the protein, our artificial spinning processes just aren't as good. The fibers are inferior. We haven't mastered the spider's elegant trick.
So we've gone from these simple color-changing tests all the way up to a material stronger than steel, all based on how these molecules are built.
From simple indicators to the complex folding of beta-pleated sheets.
What's the big takeaway from all this? For me, it's how incredibly important that primary structure, that initial sequence of amino acids really is. A tiny mistake there can be catastrophic.
You're talking about sickle cell anemia.
Exactly. The most famous example, it's a devastating disease caused by a change in a single amino acid in the entire hemoglobin protein.
At one specific position, number 6 on the beta chain, a negatively charged glutamic acid is swapped out for hydrophobic valine. That's it. That's the entire genetic mistake.
And the result is a complete structural failure. That one little change causes the hemoglobin molecules, when they're not carrying oxygen, to clump together, to polymerize into these long, rigid rods.
Which physically distorts the red blood cells from their normal disc shape into that crescent or sickle shape. They get stuck in small blood vessels, causing immense pain and organ damage.
All from one wrong amino acid. Which brings us to a final, really provocative thought.
Given how harmful this mutation is, why is it still so common in certain populations? Why hasn't natural selection just gotten rid of it?
Because there's a trade-off. It turns out that people who are heterozygous, who have one normal copy of the hemoglobin gene and one sickle cell copy, have significant protection against malaria.
Right. The malaria parasite has a harder time surviving and reproducing in those slightly altered red blood cells.
So this molecular flaw, this potentially fatal mutation, persists because it provides a powerful defense against an even deadlier threat. Think about that. What other genetic variations that we see as flaws or diseases might actually be protecting us in some way from threats we don't even fully understand yet?